Hello, dear students. How are you all? I hope you all are good, right? So, today I have brought something for you very special. Uh, I want to ask one question first before beginning this. So, which is the most difficult chapter for you in class 12th in botany, right? So, you will tell me one answer only, I know. Some of you can give me another answer, but most of you will give me one answer, that is molecular basis of inheritance, right? That chapter seems quite difficult for majority of the students. The reason behind it is that the chapter is very much conceptual. You have never seen all these things happening, right? You have never seen DNA getting formed, you have never seen RNA getting formed, you don't know what is gene regulation, so you have to imagine a lot, right? That is why this chapter is a little bit difficult, but believe me, this chapter is not that much difficult. Try to understand it, try to imagine the concepts which they are telling, have a look at some animations, have a look at some diagrams, right, where you can understand that the lines or the text is talking about what. Then you will love this chapter. So after doing that, after revising this chapter from NCRT, there are some set questions which will be asked from this chapter, right? Some of the questions I have brought up here. 10 questions per day I will be taking from this chapter. Maximum I will try to do possible questions. I will make some questions, I will gather some questions, right? I will refer some questions, and I will be presenting those questions for you. So first question, you can see, as you can see, HGP is there, right? HGP is the topic which most of the students do not like. What is the reason behind this? Because in this topic you have a lot of information to retain, right? You have to learn, right? Learn and memorize that information. Based on the small information that is given in NCRT was salient features of human genome project. After doing the entire sequencing of our DNA, we have obtained some features of our DNA, right? Entire DNA or entire genome, better to call it genome, right? That all are mentioned in some points in NCRT. Based on that, they have given this question. So you have to just find the incorrect statement, okay, with respect to the salient features of human genome project. So just quickly have a look at all these four statements and tell me what is the answer. Do you really need to memorize anything for this? If you have understood the concept of human genome project, then also you can do this, right? Quickly tell me the answer. Okay, so I think you all have understood that the answer is option number 2, right? Why answer is option number 2? Why? Okay, because there is a you can say statement is wrong, because the correct statement is less than less than 2% of genome, right? After doing entire sequencing of our genome, we understood this, that less than 2% of genome codes for protein, right? Only less than 2%. Or next time you can get a question where they will write more than more than 98% of genome does not code for protein. You tell me the meaning of both the statement is same or not, right? So in any way they represent, okay, these two will be correct, but here they have mentioned it wrong. Here they have mentioned more, right? It is less than, right? After obtaining the entire sequence of our DNA, or you can say our genome, we have got some salient features which are represented in NCRT like this. You can quickly pause and revise it, right? You can see that they have mentioned that human genome has almost 3167 sorry 316 4.7 million base pairs, right? Which they have mentioned as haploid content of a human genome is 3.3 into 10 raised to power 9 base pair, if you remember, right? In the beginning of the chapter they have mentioned average size of a gene is 3000, right? But if you consider the largest gene, that is dystrophin, and it is having 2.4 million bases, and the smallest gene is not given in NCRT, but its name is testis determining factor, or TDF, whose number of bases doesn't make any relevance for NEET, but yes, smallest gene is TDF, okay? TDF, testis determining factor. And this gene is located on chromosome uh, Y. On Y chromosome you have this gene which is called testis determining factor. This is the smallest gene, right? Smallest gene. Okay, this is not given in NCERT. Okay, now after that they have mentioned that estimated number of genes were 80,000 to 1,40,000, but we have got approximately 30,000. That is one of the statement in your question also. Out of all the, you can say, nucleotide bases which are present in the DNA, 99.9% is exactly same in all people, right? Only 0.1 differences are there, right? And 50% of the genes which are discovered, we don't have any function known for that, right? It is still under discovery. We will discover it, what they are doing, but still we did not understood what are the functions of those 50% genes which are discovered. Less than 2% of the genes they code for protein, which was our answer. They have mentioned more than 2%. And then after that, uh, majority of our genome is having repetitive DNA sequences, right? Where you have a repeating unit, like for example, AGCT, 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 it will repeat multiple times, multiple times. This kind of condition is there in your genome. And these sequences are very important because they directly have roles in mutation. They have role in changing the structure of chromosome. They are responsible for evolution also. So many things are there for them, but definitely most of them, they are having no direct coding functions, right? And after that, there are 1.4 million location where our genome is having difference of only one single nucleotide. That is called single nucleotide polymorphism. Polymorphism is a term which is used to explain what? It is used to explain the same DNA fragment, okay, the same portion of your DNA, you can say, how it is different in different people, right? If I take this particular DNA, right, same part of the DNA if I consider, okay, let's suppose this DNA is present chromosome number 1, right, and I am looking at this segment, it will be having some sequence, right? So same DNA, if I will look chromosome number when in any other person, everything will be same. Only difference will be at 1.1 base will be different. Rest all everything is same, right? This is called SNP, single nucleotide polymorphism, right? So this we have to remember, these salient features of DNA we need to study. Okay, understood. Now we have second question. You can see here, in an eukaryotic mRNA molecule, 2.4 million bases are found, right? The number of phosphodiester bonds and hydrogen bonds present in it. So in this kind of questions, you have to be very careful and you have to see that this is not DNA. They are talking about what? They are talking about eukaryotic mRNA. If they are talking about eukaryotic mRNA, definitely mRNA is single stranded, right? Not double stranded. For making double-stranded nucleic acid, you require hydrogen bonds. For single strand, hydrogen bonds are not at all required. So if hydrogen bonds are not required, then we should have how many hydrogen bonds here? We should have zero hydrogen bonds, right? Only one such option you can see or not. This is such an easy question, right? Then after that, if you have 2.4 million bases in your RNA, how many phosphodiester bonds are required to connect it? You can understand it like this. Let's suppose this is 1, 2, 3, 4, 5. Okay, these are 5 ribonucleotide. Ribonucleotides. For connecting these five ribonucleotide, how many phosphodiester bonds are required? 1, 2, 3, 4, right? 4. Okay, 4 are required. Okay, so if you have 2.4 million bases, okay, you require almost 2.4. Approximately, I'm writing 2.4 million phosphodiester bonds also, right? Okay, so 1, 2, 3, 4, 5 are there, right? So you'll be thinking 4. No, they have not given any such, you can say, option. So approximately 2.4 million phosphodiester bonds are required, right? So that's why option number 3 will be the answer or not. These many phosphodiester bonds are needed, right? So such an easy question you can see. Okay, then third question. Okay, this is a name of a bacteriophage. Okay, there are different type of bacteriophages which are given in this chapter. So this bacteriophage is 5X174. Okay, this is a virus. Okay, this has how many nucleotides? Okay, in NCRT, there's one paragraph in which they have given the number of nucleotides in E. coli in this virus, right? In lambda phage, in human genome, how many base pairs are there? So you have to learn that data. This is a database question, right? So you have to learn it. There's not such kind of concept here. You have to learn it, right? So you will see that they have mentioned that it is having 5, 3, 8, 6 nucleotides. Nucleotides, or you can even write bases. Okay, you can write nucleotides or bases. Why I'm writing nucleotide and bases, not base pair? Can anyone tell me? And if you know, you can comment below. Which of the genetic material is single-stranded DNA here? Right? It is not double-stranded DNA. If we have two strands in DNA, then only we'll be having base pairs, right? But I'm saying that we are having a single stranded DNA, not double stranded. So do we need any base pair? No. In double stranded DNA only we will use the term base pair. Okay, here only base. Okay, here one base will be there, here one base will be there, here one base will be there. Here base, two different bases will be there on two different strands, and they will be connected together with the help of hydrogen bonds, right? That's why they are called base pairs, right? Here only bases are there. So how many glycosidic bonds are required and how many phosphodiester bonds are required? What's the meaning of glycosidic bond? Glycosidic bond is a bond between a sugar and the base, nitrogen base, right? So if I have 5386 nucleotides, okay, 5386 nucleotides, then total how many glycosidic bonds are required? Same number, right? Just for example, if you have 1, 2, 3, 4, 4 nucleotide here. So for making this 4 nucleotide, you require 4 bases or not? And for connecting these 4 bases, you require 4 glycosidic bonds or not, right? So glycosidic bonds will be how many? Same as the number of nucleotides. Okay. Phosphodiester bond, just now in the previous question I told you, same number of phosphodiester bond also, right? 5, 3, 8, 6, right? So this will be the answer. Option number 2, right? Easy question or not? Okay. Can you choose any other option here? No, right? It will be other jumbled words. Okay. Now, option, this question number 4. In question number 4, you can see they have mentioned that, okay, correct the statement carefully and choose the correct option with respect to one strand of DNA. Okay, with respect to one strand of DNA, you have to give the correct answer. Okay, you have a double stranded DNA mostly. Here they are asking for one strand. What is the correct one? Okay, phosphodiester moiety in 5' and gives acidic nature to nucleotide or not? Is this statement true? Yes, always remember that the phosphodiester moiety which they are telling here. Who is that phosphodiester? What is that phosphodiester moiety here? They are talking about phosphate group. Okay. And maybe in chemistry you have learned phosphate group imparts acidic nature to DNA, right? In biomolecules, in chemistry you might have learned it. So phosphate group actually comes from where? It comes from phosphoric acid. Okay, and this phosphate group itself imparts acidic nature to DNA. Provides acidic nature. Okay, fine. 3' end of nucleotide chain contains free 3' OH group. If you consider 3' end of a polynucleotide chain, does it have a free 3' OH free group? Yes. So first statement is true. Second statement is also true or not? Okay. If we make a diagram of DNA, okay, let's suppose this is 5' position, and at 5' position you have a phosphate, right? Then you will have the sugar or not? Deoxyribose sugar, right? To this first carbon, let's suppose you are adding adenine with the help of N-glycosidic bond. Here you are adding phosphate with the help of phosphoester bond, and here you have a 3 prime OH free. This is the first position, second position, third carbon, fourth carbon, fifth carbon, right? That's why at fifth you have that phosphoester bond present there, right? By which phosphate is attached. So if I want to add a new nucleotide, I can add a new nucleotide from this 3 prime OH group, right? And here there will be a bond formation which will be called phosphodiester bond. Again, you have a 3 prime free group, right? So in every time you can see this 3 prime free OH will be present on a growing polynucleotide chain. That's why you will call the polarity also this is 5 prime to 3 prime direction. So this the given statement is correct only. 3 prime end of the polynucleotide chain contains a free 3 prime OH group. That's why it is called 3 prime end, right? Now, nitrogenous bases are projected towards outside of the DNA helix. No, right? It is absolutely wrong. If I draw here a very simple type of diagram, you have phosphate, sugar like this. This is our 5 prime end, this is our 3 prime end, right? And here you have sugar, phosphate like this. And this is 5 prime end, and this will be having 3 prime OH which is present there, right? And the bases will be projected like this. Okay, bases will be projected towards the inner side. Let's suppose here I have adenine, so what I should have here? Thymine, right? Let's suppose here I have cytosine, so what I should have here? Guanine. Let's suppose here I have again thymine, then what I should have here? Adenine, right? And in between these, what should be there? Hydrogen bonds, 2 hydrogen bond between the adenine, thymine, and 3 between the guanine and cytosine. So this is how the DNA is, right? If you don't want to see this kind of structure, you can imagine that it is like this, right? In this also, it will be similar. If you want to draw, imagine it like a cross, you can imagine like that also, right? But the condition is like this. So you just see option number C, nitrogenous bases are projected outside. No, they are projected towards the inside. This is absolutely false statement which they have written, right? This is not correct, false. Phosphate moiety is projected towards inner side and the pentose is projected towards the outside. Now you understood this statement is absolutely false. If this is the diagram, then how we can say that this statement is correct? Phosphate is not projected towards the inner side and pentose is not projected towards the outside. Phosphate and sugar make the backbone of DNA, right? And nitrogenous bases are projecting towards the inner side. Okay. So what is correct here? A and B is correct. Option number 1, right? Now, here in this, just see, they have given one question here. Find the incorrect match. Okay, you have to find what is wrong here. And here some options you can see which are not provided in NCERT, right? So some outside questions can be there. Like here you can see that nucleoside is basic in nature. Is this correct? What is the meaning of nucleoside? First you tell me in the comments. Nucleoside means they are talking about a sugar, right? Plus base. Sugar plus base makes what? It makes nucleoside. In this nucleoside, if I attach a phosphate, in this nucleoside, to this nucleoside, if I add a phosphate, right? What I will be able to make? I will be able to make nucleotide. Now tell me, can I say nucleoside is basic in nature? Yes, absolutely we can say. Phosphate is not attached. Okay, this gives acidic behavior, this gives basic behavior, right? Okay, so it is correct only. Purines, okay, purines are bonded to the sugar by the ninth position. Dekho. Always remember, sugar's first carbon only will be used for attaching the base. जैसे कि for example, if this is our like pentose sugar, okay, where's the first carbon in this? This is first carbon, right? This is second carbon, this is third carbon, this is fourth carbon, and this is fifth carbon, right? So you are going to attach your base, nitrogenous base, only always at this first carbon of the sugar. So first carbon of the sugar always interact with the base. This is the basic thing which you must know, right? Fifth carbon always interact with the phosphate. Fifth carbon of the sugar always attaches to phosphate. Okay, this you have to study. But in NCERT, they have not mentioned which position from a base will be interacting to first carbon of the sugar. So here, if you see, uh, in case of purines, okay, what are purines? Purines you all know, adenine and guanine are purines. And what are pyrimidines? Pyrimidines are, okay, thymine, cytosine and uracil, right? So when it is purine, ninth position will always be attached to the first carbon of the sugar, right? So ninth position, can you see in this diagram? This all is not there in NCERT. Okay, so don't get worried. This is the ninth position. This and this, okay, in adenine, ninth position is here. In guanine, ninth position here, right? Some differences are there only. Ninth, nitrogen is there. This will interact with the, you can say, carbon, first carbon of the sugar, right? In pyrimidine, it is not ninth, it is third position, right? Pyrimidine, you can see third position. Third position pe also nitrogen is there. That will attach with the carbon of the sugar, right? So you can see very well here. Okay, I marked here uracil. I'm sorry, it should be thymine. Okay, it can be uracil in some cases, but normally it will not base pair anywhere. It will base pair in RNA only, not in DNA, right? So that is the condition here. Then if you check this, so it means that these three statements are correct only, right? Then what's the answer? Answer is asking what is wrong. So you got the wrong answer, right? Wrong answer is option number 4. This statement is NCERT statement. So do not think that this question is out of NCERT. No, you have to eliminate and get the answer which is given in your NCERT. Okay, if you check here, just see the diagrams of uracil and thymine and tell me what is different here. If you see the diagram of thymine, okay, you will see that there at fifth carbon, can you see any unusual group attached? Is that group present in that also? Rest all you check, everything is absolutely same, right? Okay, only difference is this. What is the meaning of this difference? Difference is, we can say that 5, okay, 5, we can say 5 methyl, 5 methyl uracil equals to thymine. Okay, this you need to learn. Okay, what you need to learn? You need to learn that if you attach methyl group at the fifth position in uracil, you will be making thymine, right? And what they have mentioned, uracil has methyl group at 5' position. Does it have anything? No. But if you attach a methyl group, that uracil will become thymine, right? So question was on this, and this is given in NCERT. This is not out of NCERT question. This is given in NCERT. Okay. Okay. A new concept which we have to learn is purines always connected from their ninth position with the sugar. Sugar always interact with the first carbon, right? To it, ninth position of the purine will attach, and pyrimidine, third position of the pyrimidine will be interacting with first position in the sugar. Okay, so one more question you understood. This is such an easy question. Tell me the answer very correctly. What material will not be used for DNA fingerprinting or DNA profiling? Can we use saliva, sperm? Can we use bone? Yes, but what we cannot use is mature RBC. What's the answer? Why? Why? What is the reason behind this? You tell me. In the mature RBC, okay, do you have nucleus? No, nucleus is absent. Okay, nucleus is absent, right? If nucleus is absent, then will you get any DNA? No, DNA is absent. And if DNA is absent, can you do DNA fingerprinting? Absolutely not, right? So if you are getting any kind of cell which does not have nucleus or which lacks nucleus at maturity, or it means that it cannot be used for DNA fingerprinting. While saliva, sperm, bones skin cells, any other cell which is a source of DNA, where you can extract DNA, where you can isolate DNA, you can do this process, right? So option number 3 will be the answer for this question. Now one question you can see, recognition sequence for transcription in bacteria. Again, you will say this question is not in NCERT, but last year in NEET 2024, they have asked one question, answer was there in the match the following. Even if you don't know, you can solve that question. That is very easy question. You can go and check it back. Okay, still I have added this so that you can learn, you can know what is the meaning of recognition sequences, right? So if I talk about recognition sequences, these are very important sequences. These are, first of all, these are conserved sequences. Okay, cannot be changed. These are conserved sequences present in DNA, which acts like recognition sites. Okay, which acts like recognition sites for RNA polymerase to bind. Okay, bind on correct promoter. Understood? So you know DNA after getting formed, if you want to make any RNA from that DNA, you need to do transcription, right? RNA formation or RNA synthesis from DNA is called transcription. And for that, you require a very important enzyme which is called RNA polymerase, DNA dependent RNA polymerase. This RNA polymerase will go and search the site where to bind on the DNA to start the process of transcription. This binding site will be, you can say, recognized by some specific recognition sequences which are present near to the promoter. Promoter is a site where RNA polymerase will bind, right? A promoter, how the RNA polymerase will know where is the correct promoter? Promoter, there are some specific recognition sequences which are present here. So I have given one image here so that you can understand. This is a bacterial transcription unit. And then in this bacterial transcription unit, you can see +1. This is given in your NCERT +1, right? +1 means transcription start site. From here, transcription has to start, right? It means that before transcription start, this is promoter, right? In this promoter sequences, in this promoter, there are certain conserved sequences which are present, which will be searched by RNA polymerase, right? अब देखो, the place, or understand the place from where the transcription will begin, it is transcription start site. Okay, before this, whatever is present is called upstream. Okay, whatever sequences are present here, okay, they are called upstream of transcription start site. After this, whatever sequences are there, they are called downstream, right? Understood? Upstream means before transcription start site, upstream means before, and this is after transcription start site, these sequences. Okay, sequences which are before transcription start site, they are, you can see, if I get the position, if this is +1, right? If we go before this, it will be 0, then -1, -2, -3, -4, -5, like this. And if we go after +1, so +2, +3, okay, like this it will be. Okay, so at the -10 region, you will be having some specific consensus or conservative sequences which are called Tata box, right? This is called Tata box. Why? Because this sequence is rich in TA. Okay, and then one more box is there which is called Pribno box. Okay, Pribno box, whose sequence is TTGACA. These are the two sequences. One as minus, this is also called -10 box, -35 box, right? These sequences will be searched by RNA polymerase so that it can bind on it, right? Because these are promoter sequences. Okay, in the promoter, there are some specific sequences which are searched by RNA polymerase to allow binding, right? So these two sequences are not given in NCERT, but they have given this as an option in match the following in 2024 paper. You can check that, right? So which option is matching here? Recognition sequence for transcription in bacteria, is it TATAT or TATAAT and two sequences C I have mentioned same. Okay, so it is TATAAT, right? So while typing I have done a mistake, right? So anyways, this will be the answer. Okay, you can consider this if I am writing, let's suppose C, right? C. Okay, you can correct it. Okay, so if it is C, it will not be. It is a specific sequence which should not be changed. TATAAT, right? Or instead of this, if they are writing TTGACA, then also it is fine. You have to learn this. You cannot change this, right? So that is there. What is these sequences? They are searched by RNA polymerase to bind. They are specific sequences which are present in the promoter region so that your RNA polymerase can bind correctly to the promoter. If you consider where these are present, they are present -35 and -10 region from the transcription start site, okay, which is before the transcription start site where promoter is there. Okay. Now this question. In Griffith experiment, heat killed S strain transfers what to living R strain to make what, right? So you know Griffith's transformation experiment principle, right? It is very, very easy. Griffith has used one bacteria. What is the name of that bacteria? Bacteria he has taken was pneumococcus, or you can say streptococcus. NCRT, I think streptococcus is mentioned, right? Streptococcus pneumoniae. Okay, this is streptococcus bacteria he has taken, streptococcus pneumoniae. It was having two strains, right? Two strains. One strain is virulent strain. Virulent. Virulent means it can cause disease. Which disease this causes? It causes which disease? Pneumonia, as the name is telling itself, pneumonia. Right? Virulent and avirulent. Two strains are there. Virulent strain is that strain which has mucus layer. Avirulent is that which cannot make mucus. The name of the strain which is virulent, which can make mucus, actually the pathogenicity is because of mucus only. The name of that strain is S strain. And the name of this strain which cannot make mucus, it is R strain. This is also called smooth. Okay, this is also called rough. Okay, because it makes smooth colonies in culture. It is making rough colonies because it lacks that mucus layer. Remember, glycocalyx one part is uh, that layer capsule which is called, right? So that is the thing. Now what they have mentioned in Griffith experiment, heat kill S strain transfers what? They have done this experiment, right? In which they have injected the mouse with the R strain. Then nothing happened. Mouse was alive. S strain, nothing happened. Mouse was alive. Sorry, S strain when they added, then mouse got killed because it was virulent. Pneumonia occurred in that mouse. Then they added heat killed S strain. Then mouse was living because the mucus layer got damaged in that, right? And DNA also got denatured, so it cannot prepare the mucus again. Cell was also not there, right? They damaged the entire content. Now in one experiment, in the last experiment, if you remember what they have done, they have used heat killed S strain plus they have taken alive R strain. Okay, it means here capsule was absent. Okay, here also capsule was absent. So that character which was causing pathogenicity or disease was absent. But to their surprise, what happened? Mouse died. Okay, or mice died because it was experiment done on so many mouse. So mice died. So what you have to understand here, you have to understand that something passed from S strain, heat killed S strain to R strain, and it transformed this R strain into S strain. Okay, what happened? Something got transformed from S strain to R strain and converted that R strain into S strain. Okay, so S strain, R strain, transformation into S strain. How this happened? Let's suppose bacteria is capable to make capsule. Why? Because it is having that particular DNA, right? But by boiling, by heating it, what happened? The DNA got separated. DNA might got fragmented also, right? So because of this, what is going to happen? Because of this, now that particular cell cannot make capsule. Even the cell will got damaged, right? So it will not be able to make capsule. Now what we have to do? Now, if we allow them like S strain heat killed and R strain alive together if they are present, what will happen? That particular DNA which got denatured, which got single stranded, it will again, when the temperature will become normal, it will again bind back, right? If it will again bind back, it doesn't have any cell. So the R strain cell which is present, which was not boiled and heat killed, that DNA will be like it will be taken up by R strain. R strain will take the DNA of S strain. And now that S strain ka DNA will be having the genes for the sense of capsule. It will be able to make mucus layer on R strain and it will convert that R strain into S strain, right? And that is the reason that why mouse died, right? So what we have to understand, in the Griffith experiment, heat killed S strain transfers DNA to the living R strain to make it virulent. But the thing is that after the end of this experiment, they have mentioned they have not found the nature of the material which is moving from S strain to R strain. So that's why, because chemical nature was not clear, right? Chemical nature not clear. That's why they called it as transforming principle. Some transforming principle, it was moving from heat killed S strain to R strain. It was converting the R strain into S strain. Understood? Okay. Now, which character of RNA does not make it genetic material? Very, very easy question, right? Very easy question. In this question, just see monocystronic condition, polycystronic condition. So, doesn't make any sense, right? What's the meaning of monocystronic? Monocystronic means one transcription unit can make one protein. Single transcription unit can make a single protein. Polycystronic means single transcription unit can make multiple proteins. An example is lacoperon itself, right? Anyways, these two are out of the options because that does not impart anything with respect to why RNA should be considered as genetic material. This is present itself on DNA, right? Only two options you can consider: autocatalytic nature and high rate of mutation. In this, what is the reason that you will never choose RNA as genetic material as per NCERT? So it must be option number 3, right? Can you imagine that every now and then, every second, your genetic material is changing? No, right? Because if mutation is occurring, if any change is occurring at the genetic level, what will happen? Direct change will be there in your phenotype, right? So sometimes the changes can be good for you, beneficial for you. Sometimes they can be lethal for you, right? So high rate of mutation is the main reason because of which we should not, like we have not chosen RNA as our genetic material. Autocatalytic nature means RNA can make it like RNA. It is catalytic, autocatalytic, right? DNA is not autocatalytic. For making DNA, you require so many things, right? But RNA can act like a ribosome, right? DNA cannot itself do any work, right? But RNA acts like a ribosome, so it is autocatalytic in behavior. But that does not mean that we can choose it as genetic material. The reason not to choose RNA as genetic material, the most appropriate reason is that rate of mutation is very high here, that we cannot afford, right? And now this, the 10 question, again you can see this is little bit out of NCERT, but it's fine. We have to prepare for the you can say worst also, right? Because we don't know what they will ask. So how many enzymes are involved in DNA replication eukaryotic nucleus as given box? So you can see plenty of enzymes are given: topoisomerase, helicase, primase, ligase, DNA polymerase 1, 3, gyrase, and all of these things. If you want to copy eukaryotic DNA, right? So for copying eukaryotic DNA, I hope you have learned little bit more than NCERT. For opening the two helix, like your DNA is like this, right? For opening the DNA helix, you require one enzyme which is called helicase. It actually breaks the hydrogen bonds and it unzips it, right? So helicase is required. Okay, no problem in this. Then you require DNA polymerase for making, you can say, DNA. But DNA polymerase 1, 3, and 2, they are not a part of eukaryotic DNA replication. They are prokaryotic in nature. That's why DNA pol 3 and 1 I am cutting here, right? After that, you require ligase also, sorry, primase also, because you know DNA polymerase does not have any capacity to initiate the replication. It can only extend the replication if it gets 3' OH. So for that, primase will be making a primer. That primer is a small RNA molecule which will provide a 3' OH, and then DNA polymerase can extend that, right? Let's say, for example, a small RNA is created here, right? So it will give a 3' OH that will be further extended by DNA polymerase. Here you will make DNA. Okay. After that, I also told you that you have topoisomerase. While you're opening the DNA helix from this side, there will be a negative tension generated here. So you need to uncoil this portion by the help of one more, you can say, enzyme, which is called topoisomerase. So these four are needed, right? These four are needed, but here you can see uh, topoisomerase, helicase, primase, and one more I will write here, that is ligase. Okay, ligase. You know that while making the DNA, okay, let's suppose this is 5' to 3' template strand, sorry, 3' to 5' template strand, always you will see from this side, and this is 3' to 5', or better to call it from 5' to 3'. While writing, we will write 3' to 5' template, and this we will write 5' to 3'. Okay, from the open end we will consider. Now, in this one, if you consider this strand, okay, you can easily make DNA in this direction. But on the opposite strand, you cannot make easily DNA in the same direction, right? You have to make small, small primers first of all in opposite direction like this, and then you have to extend the DNA in this fashion, right? So all the primers need to be removed, and again DNA must be synthesized, right? And after the synthesis of the DNA, all the DNA fragments must be joined together, in which ligase will help, right? So I have written here 5. Out of these 5, how many you can see there? You can only see 4, right? Okay, this gyrase is just like topoisomerase, but it is there in bacteria. So here option number 2 will be the answer. Now, why I have not included DNA polymerase here? For that, you just see this table. This is not necessary for you to learn this table, but at least you should have an idea that if they give anything from here, you should at least be able to differentiate that this is not in prokaryote and this is not in eukaryotes, right? So 1, 2, and 3. These 3 different types of DNA polymerases are required in prokaryotic DNA replication, right? Some are there, like DNA polymerase 1 is for DNA repair and gap filling, okay, connecting Okazaki fragments, that is the meaning of gap filling. DNA polymerase 2, DNA repair, okay. DNA polymerase 3, main replicating enzyme. And I'm sorry, DNA polymerase 1 will be responsible for, like after the removal of primer, it will be responsible for filling the gap by making DNA again. Don't think that DNA will be joined by DNA polymerase. No, that will be done by ligase, right? After removal of primers, new DNA will be synthesized by this DNA polymerase 1, and then the fragments will be joined by ligase. Okay, so these are the 3 enzymes, and these are in eukaryotes: DNA polymerase alpha, okay, DNA polymerase gamma, okay, DNA polymerase delta, epsilon, okay, all of these are there in our cells. But you don't have to learn the entire mechanism of their work. Only just for differentiation I have mentioned it, so you can have some extra knowledge. Okay, okay, then this was all. I hope you have understood many things in this session. I will try to bring some more questions like this every now and then, right? So thank you everyone for watching. And if you have any question or query, you can write in comments. Thank you.